So I guess we can start. So hello, everyone. It's with both pleasure and profound sadness that I introduce Shubham Chandak. Shubham has become a central pillar in our group for research, for teaching, for technical and emotional support, and everything in between. You guys know the story about the man who goes to the doctor because he's stressed? And the doctor says, I recommend you start meditating for half an hour every day. And the man says, where am I going to find half an hour for this every day? And the doctor says, in that case, I recommend you make it a full hour. So this story always reminds me of Shubham. He never seems stressed, no matter how many projects and tasks he's working on. And he's always with a relaxed yes and attitude to whatever else comes his way. And while also, by the way, I recently learned, even more relevant to this story, he finds time for, and probably largely thanks to, his daily Vipassana meditation sessions. Throughout his time here, he's been pursuing, in fact, leading several research thrusts, while at the same time being central to our curricular development efforts. This quarter, for example, he's working with Ifer and me on developing and TAing a new engineering fundamentals course in conjunction with this research. So to keep things fairly concrete and focused, he'll be writing his thesis and therefore dedicating today's presentation to his work on genomic data compression. But he could easily have written theses based on his work on storage in DNA or his work on multimedia streaming. I encourage those of you curious to peruse his website for more. So without further ado, I give you Shubham.
Thank you, Tsachy, for the kind introduction. Okay, I will share my screen now. Okay. So as Tsachy mentioned, today I'm going to talk about compression of raw genomic data. So let's get started. So I will start with some introduction and motivation, and then I will talk about two of our works in this area. So let's talk about what is the genome. What do we mean by genome sequencing? Why do we even care about compression, and what does a typical genomic processing pipeline look like? Right. So as many of you might already know, your genome is the primary hereditary material that you inherit from your parents, and which your genome lies in each and every one of your cell, and it determines a lot of your physical characteristics like height, eye color, and perhaps more importantly, it also determines whether you are more prone to certain diseases, right? So it is very important for us to understand our genome. So at a very high level, the genome is just a sequence of DNA bases where each DNA base can be A, C, G, or T. So this is a simplified picture, which is enough for this talk, right? So on the right, you can look at the names of all the DNA bases and so on, as well as the double helix structure. So the DNA occurs in two complementary strands. The information in one strand is enough to determine the other strand. So you can just focus on one strand in terms of representation. And for humans, the DNA is really long. It's 3 billion bases in total, multiplied by 2 because you get one copy from each parent. And it's divided into 23 chromosomes. Right. So over the past 20 years, maybe slightly more, people have developed ways so that we can actually read this 3 billion base long string, right? So the process starts like this. You have a large DNA molecule in some sort of sample. Unfortunately, we can't really read it as a single long string of 3 billion bases or like even one chromosome in its entirety. So therefore, the processing actually goes through something called fragmentation, where you divide the long chromosomes into shorter pieces. and also amplification, which is just a way of duplicating DNA into many copies. And after that, each of these short pieces, you can read them in something called the sequencer. Hence and this process is called genomic sequencing. At the end of this process, you get something called reads. So reads are like these short substrings, which you get from the larger DNA molecule. And then you can use these reads in various ways, as we will see shortly, right? So one thing to note here is that even though your DNA may be of length 3 billion, the total length of all the reads can be much longer, because we have this amplification step, and we actually get data which is much more in volume as compared to the original genome. And this has been really facilitated by the rapidly dropping cost of genome sequencing. So in around 2001, the cost of sequencing one genome was around $100 million, whereas today it's lower than $1,000, right? And it's even faster than the Moore's law, which determines how most of the computing technologies progress. So in some sense, the cost of genome sequencing is dropping faster than basically all the analysis tools and the storage tools. And this has led to really large projects. So here we look at just a few of the big projects that are either going on right now or planned for the near future. So we have the UK Biobank project, where they plan to sequence 500,000 human genomes. We have the Genome Asia project, where they plan to do 100,000 genomes. There is the Million Veterans project, where in the USA. And it's not just about humans. Of course, humans are important because we want to understand a lot about human diseases and how can we prevent them, and how can we diagnose them using genomics. But it's also about all the other species on Earth. We want to understand their diversity, and it's particularly useful in the context of agriculture. So therefore, there is the Earth Biogenome project, where they plan to sequence 1.5 million eukaryote species. So as you can guess, even from one sequencing experiment, you get a lot of data, but through all these experiments, the amount of data is just astronomical. And according to NHGRI, a US government body, over the next decade, we expect around 40 exabytes of data from the genomic projects, and 1 exabyte is a billion gigabytes. So it's going to be really large. So clearly, efficient ways of storing it, efficient ways of transferring it, of processing it is really important, right? So before we talk about that, let's quickly try to understand how we actually work with this genomic data. So ultimately, the aim usually is that you have these sequenced reads, which, as you saw earlier, are just shorter substrings of the genome, and we want to use it to learn about the original genome, right? So there are two major analysis pipelines, and we will look at them one by one. So first we look at something called genome assembly. So the idea behind genome assembly is that from these shorter substrings, you want to somehow fit them together to get your original genome. And so the first big project in this direction for humans was the Human Genome Project, which took about a decade. And they for the first time, they actually assembled the whole human genome. And you can think of it as like solving a puzzle in some sense. You have a lot of these reads, you try to find these reads which overlap with each other, and that gives you a longer and longer substrings, and ultimately you can get the entire chromosome. But it's a very computationally expensive process, and we don't want to do it for each and every human. So here comes the nice thing. In fact, for any two random humans, their genome is 99% similar. So you don't need to repeat all your efforts for each and every person. You can save a lot of effort by something called alignment. So here the idea is we start with a reference genome. So a reference genome you can think of as a genome for some typical person. So it's just some genome from some person. You take the reads from your genome and you match the reads to the reference genome, right? So you find out where the read occurs in the reference genome. And since your genome and this reference genome is around 99% similar, most of the reads you can find out where they match. And then after you do this, you look at maybe you look at this position and you see, okay, like the reference has an A at that position, but all the reads have a G at that position. So you say, okay, so this is a variant, this is a mutant, this is basically the reference had an A and instead you have a G. And ultimately, this is really important because this actually tells you that, oh, my genome differs from the reference genome in these positions, and then you can use this to predict diseases and all sorts of things, right? So this is the so-called alignment and then variant calling pipeline. And as I showed before, the cost of sequencing has really dropped over the past 10, 20 years. And it is due to like various sequencing technologies. So let's look at two of them. So one of them is the short read technology called the Illumina technology. And we show a representative device here. So you can put it on your disk or something. So this is a really high throughput. You get a lot of data. But unfortunately, the sequences you get are quite short. They're around 100 to 150 length as compared to the genome, which is like 3 billion length long, right? And it's very accurate. So this has some nice advantages. It's very high throughput and it's very accurate. But more recently, there has also been a lot of interest in new technologies. For example, the Oxford Nanopore technology, which has some nice advantages. It is really portable. It's only as big as a mobile device. It's real time. You start getting the data as soon as you put in your sample. And perhaps the most important advantage is the long reads. So this can actually give you hundreds of thousands of bases or millions of bases long reads, which makes a lot of the analysis pipelines really easy. It has higher error rate, but it also has some other advantages like native DNA and direct RNA sequencing. So overall, both of these technologies are being used heavily today. And we will talk about data compression techniques for both of these technologies. So I see some questions on chat, but I will take, I think, questions at the end. Yeah. So, okay. So with that motivation, we understand that genomic data is important. We understand that compression is important because it's a lot of data. We look at our first work, which is Spring, a compressor for FastQ data, and we will learn what FastQ means shortly. So this is joint work with Kedar, Idoya, Mikhail, and Tsachy. Right. So to talk about Spring, we first need to understand something called paired and genome sequencing, which is the default mode usually for Illumina sequencing. So again, as I mentioned before, you can think of genome as just a long string of bases, of 3 billion bases in the case of human. And the genome is sequenced as noisy paired substrings, which we call reads. So the way the sequencing works is that from the genome, you get these fragments, and then you read the left part and the right part of the fragment. So this is like a blue and green. So blue is the left part, green is the right part, and you get this as a pair. And in this figure, you can see, okay, like you get this read pair from the first part of the genome. The overall length of the read pair is around 300 to 500 bases long. And each of the read in the pair is around 100 to 150 bases long. Like any biological, any chemical, any basically physical process, it's not perfect. So you see these red dots, which denote that there are errors, right? So these are not perfect substrings of the genome. These are noisy substrings of the genome. And the typical form of error for Illumina sequencing is just substitutions. So instead of a C, you might have a A. Instead of a G, you might have a T, that sort of thing, right? And one important metric, which we will talk about again and again, so it's important to talk about it now. It's something called coverage or depth. So as I mentioned before, when you sequence, usually you sequence data from a lot of different cells, and it is further amplified during the procedure. So it's not like you are reading the genome only once, you are reading the genome many, many times. And as you can see here, even at a single position on the genome, you have a large number of reads. And so the average number of reads at any given position on the genome is called the coverage or depth. And for a lot of applications, it's around 30 to 60x. Sometimes it's higher for some applications, but we will take this as the typical value. Right. So as we saw before, when we do sequencing, these raw reads are just the raw product, raw material in some sense. It is followed by a lot of analysis. So why do we care about storing the raw reads? Why don't we just do the assembly and store the assembled genome? Or why don't we do the alignment variant calling and just store where our data differs from the reference genome? So there are a few reasons, right? So one reason is that pipelines, all these analysis pipelines improve with time, and we need raw data for analyzing it again and again. Because if we throw it away, then we can't do it anymore. Another really important reason in practice is that you need to often store it temporarily, or the government regulations require you to store it for some period of time. And finally, there are certain applications when you don't have a reference genome, and in those cases, again, you need to store the raw data because it is your primary source of information. Right. So now we are convinced that storing raw data is important. So let's look at how the raw data is stored. So it is typically stored in something called the FASTQ format, and we will look at a typical file here. So as we saw earlier, the data is stored as paired reads. So that's why it comes in two files. So file 1 and file 2. In each file, you have like multiple reads. So each read is a substring, right? And the first read in file 1 is paired to the first read in file 2, and so on. So you see the read sequence here in green, and you see the ACGT and so on here. But you also see some other information. So in blue, you see something called the read identifier, which is just metadata about the read. It tells you where the read occurs in the sequencing machine and all that sort of information, some sort of unique ID for each read. And you also have something called the quality score, which is just a character representation of how confident the sequencer was when it said that this is A, right? Like, was it very sure that this was an A, or was it like not so sure, right? So all this is based on a physical process, right? Like a chemical or physical process. So it has some confidence scores basically. In this talk, we will mostly talk about reads and very quickly talk about quality and read identifiers at the very end. So, okay, so let's look at read compression. So let's think of a typical 25x human data set. So 25x means that, okay, you have a human genome of length 3 billion. You are reading it 25 times. So it's around 75 billion. And if you store it uncompressed, the total size is around 75 billion. So it's around 79 billion bytes. So 79 gigabytes. And this is because we store it just as a character. So ACGT, each one of them takes 1 byte. This is clearly very inefficient, and you can just use something like Gzip, which is a very standard compressor, and it will bring you down to 4 times lower, so around 20 gigabytes. And this is 2 bits per base. So you have 4 possible bases, A, C, G, T. So A can be represented as 00, C as 01, and so on, right? But the question arises, is this optimal? And actually it's not optimal. And why is it not optimal? Because as we saw in the previous figure, you are reading the genome many times. So clearly there is a lot of overlap between all these different reads. Like you have a lot of reads which have the exact same sequence or like roughly the same sequence. So we should be able to use that information to improve our compression. And indeed we can, as we will see next. So before that, another thing that might help us is that, as we saw, we have these pairs in the FASTQ files. But how about we reorder the pairs? Like, can we reorder the pairs? And the answer is yes, because the pairs, the order of these does not tell you anything useful. It's just some random ordering due to the order in which they were read from the machine. So you can reorder it, and it can save you a lot of space, because, for example, you can put reads that look similar closer to each other, and you can put things that are not so similar far from each other. Okay, so as we saw, uncompressed was 79 gigabytes, Gzip was 20 gigabytes. So FastStore, which was a specialized compressor before Spring, and if you allow like the reordering of the pairs, as we saw in the last slide, you can get down from 20 gigabytes to just 6 gigabytes. So which is huge improvement over Gzip, which was already compressed. And our work, Spring. So Spring, if you allow the reordering, you can get from 6 gigabytes to further 3 times reduction, which is 2 gigabytes. As you can see, it's like quite good. It's 10 times better than Gzip. And even if you want lossless, for some reason, you want to store the file as it is exactly, you want to keep the order intact, you still get 3 gigabytes, which is still quite a bit better than Gzip fast store uncompressed everything. And it's not only like this data set, like if you take a much larger data set, so this is a 319 gigabyte data set when uncompressed. And you can see Spring still gets it down to 10 gigabytes, which is 8 times better than Gzip. And if you allow reordering, it's even better. So we will clearly see that like if we can do it smartly rather than using a general purpose compressor, if we use something specialized, we can save a lot of space. And we will see later that it's slower than Gzip, but it's not too slow. It's still practical. So let's look at some key ideas that went behind the design of Spring. So our first intuition came from a theoretical analysis. We were just trying to understand, if you are trying to store reads, what are you really storing? So this is a thought experiment. Let's say we know the genome from somewhere, like we know the true genome. Let's say we know where the reads, for each of the reads, we know where it starts on the genome. So these yellow, like for example, this read starts at position 6, let's say. And we know for each pair, how far apart are the paired reads. And let's say we also know where the noise, like where does each of the reads differ from the actual genome. So suppose I know all these 3 things. This is sufficient to reconstruct all my reads, because I can just take the genome, I can take the appropriate substrings, and I can add the appropriate noise. So this is enough to store the reads. And as you can see, like if you have multiple reads at the same position of the genome, you are not storing the data again and again, because you're storing it just once when you store the genome. So this is clearly more efficient than like just listing all the reads one by one. And indeed, we did some theoretical calculations using the entropy, and we saw that this can do much better than Gzip, of course, but even previous compressors. So we found that the previous compressors were not doing things optimally. So there was some scope for improvement. Of course, it's not as easy because we don't know the true genome. We just have the reads, we just have the FASTQ file. So how do we get the genome from the reads? You might say, okay, do the assembly. We already saw we can assemble the genome by fitting the puzzle in some sense. But this is a very expensive process, and we don't want to do genome assembly just for doing compression. And there are a lot of challenges, like the genome has a lot of repeats. It's very hard to assemble in general. And here comes like the key idea that we don't need perfect assembly for compression. If we can do some sort of greedy approximate assembly, as long as we can do good compression, that's all we care about. And we can get like 10 times faster, even 100 times faster than the assembly algorithms just by doing it greedily, and where our only focus is on compression. So with that, the workflow is relatively easy to understand. We start with the reads. We do this approximate assembly process, and this is sort of the core of Spring. I won't go into the details now, but happy to talk offline. But ultimately, it's just some sort of greedy assembly procedure, which is really efficient. It is designed to be really efficient. And what it does is it finds reads that overlap with each other, like reads which have some common substrings. And it basically like compiles them together. And using these reads, we get these green sequences, which are sort of like the chromosomes or like assembled sequences. And we find out where these reads differ from our consensus or our assembled sequence. And once we have this assembly, we can encode it. We can store the assembled sequence shown in green. We can store where each of the reads starts in the assembled sequence. We can store the gap between the paired reads. We can store all the noise, like where does the noise occur, what is the noise, and so on. And once we have these, it's really easy to compress them with any general purpose compressor. We could use Gzip, for example, but we use BSC, which is basically a much better compressor and still quite efficient. And this just gives us the compressed file. So this is as simple as that. This is the workflow for Spring. And really like the big contribution here is in the approximate assembly step, where we used a hash-based technique to make it very efficient. And one final note. So in the allow reordering mode, like where we allow the reordering of the pairs, the only change is in this one, the read position in the assembled sequence. And what we do is we just sort our reads according to the position in the assembled sequence. And that can save like a substantial amount of space. All right, so let's look at very briefly at quality and read identifier compression. So quality, as we saw before, quality was just denoting how much each of the, like what is the confidence for each of the ACGT bases, right? And here we just use a general purpose compressor, BSC, and it gives quite good results. And we Spring also has a lot of modes. You can like do lossy compression, lossless compression. We won't go into that, but basically you can do a lot. Read identifiers. So here we just use an existing solution, which is quite commonly used. And it's just based on, basically you split your thing into tokens, and each of the token you compress independently using arithmetic coding, which is a very standard technique in compression. Right, and each of the tokens sort of like if you look at consecutive reads, the tokens, most of the tokens remain the same. So it is very, very compressible. And we can actually see that here. So here what we are trying to see is how much do each of these three components, so reads, qualities, and read identifiers, how much they contribute to the compressed file. So after compression, how much is their relative size? And we look at two data sets. So they're mostly the same. Both are human data sets. Only difference is that they were sequenced using different machines. So the top one was using an older HiSeq 2000 machine, and this one was a newer NovaSeq machine. And okay, so you see that the read identifier, the yellow one, is really small. So after compression, read identifier is a very small contributor. The quality for the older machine, for HiSeq 2000, is really, really big. So quality values used to take up a lot of space even after compression, and therefore there was a lot of focus on how to compress these efficiently. But what happened was, like at some point, Illumina decided that we can really, like earlier they used to use 40 different levels for denoting the quality. Then they went down to just 4 levels, and that was good enough. And so basically today quality values are a very small contributor, and therefore we mostly focused on the read compression, because like basically any improvement in read compression is a substantial improvement to the overall file size as well. Right. So Spring was not the first compressor for FastQ files. There have been a lot of compressors over the past, but unfortunately, most of them don't get much adoption. Everybody still uses Gzip. So therefore, our aim when we built Spring was to make it as practical as possible, so people actually use it. It's just not like some academic work, right? So let's look at Spring in action. So we could start with a 195 gigabyte 25x human data. In 2 hours with 32 GB RAM, and Spring is multi-threaded. So with 8 threads, you can get down from 195 gigabytes to just 7 gigabytes. Whereas Gzip would give you 36 gigabytes, FastOver would give you 11 gigabytes. In terms of computation, Spring is comparable or better than other specialized compressors. It's slower than Gzip, definitely. Gzip takes around half an hour for this data set with 8 threads. But it's not too bad given the amount of space we are saving. And decompression, which is much faster, you can do decompression and you get exactly the file you want. So Spring supports lossless compression. It matches the original file byte by byte. And Spring is really easy to use with support for lossless modes, lossy modes. You can reorder the read pairs. You can do lossy quality value compression. So there are a lot of things I didn't talk about, but Spring supports everything essentially. We made Spring so that it supports all types of non-idealities in the original file. So in general, it just supports, like you give it a FastQ file, Spring will compress it. Spring compresses the data in blocks, which allows us to do streaming decompression. So you can start processing your data even as you are decompressing it, right? It is scalable to large data sets. We tested on a lot of different organisms apart from humans, and in general, it works well. And Spring also supports Gzipped input output. So you don't need to, like if your original FastQ file is already Gzipped, Spring can take that as the input directly. Right. So some of these, of course, like Gzip also supports, but a lot of these are because Spring is a specialized compressor for FastQ data, and Spring is open source, available on GitHub. right? So it has been around 2.5 years since Spring was published. And so let's look at where we are right now. So Spring has been downloaded more than 1,500 times from Conda, which is a popular library for like open source software. There has been some interest from industry and some medical institutions who contacted us. They wanted to add additional features to Spring. They wanted to adopt Spring. And there has, of course, people have been working on other compressors after Spring, but the nice thing is that a lot of them use the overall same paradigm as Spring. And some of them improve the compression at the cost of computational requirements. So clearly there is some further scope for improvement. But the general paradigm has roughly remained the same after Spring. To further increase the adoption, we actually also are also integrating Spring as part of the MPEG-G codec, which is a way of standardizing genome compression. It's like MP4 or MP3 for audio video. MPEG-G is the one for genomic compression. And Spring is part of this standard, and we are working on integrating it into an open source codec for this standard. And finally, we are currently working on extending the ideas from Spring for to long reads, nanopore data with other challenges. Right. Okay, so with that, let's move on to some lossy compression for nanopore raw signal data, so which is the second part of this talk. So this is joint work with Kedar, Srivatsan, and Tsachy. Right. So this is a shift of pace. Earlier we were talking about Illumina sequencing. Now we will talk about nanopore sequencing, which as I mentioned earlier, is a new sequencing technology which allows really, really long reads and a portable device like a mobile phone. So as the name suggests, you see a pore here. So nanopore, pore, right? So the pore goes across a membrane. So the horizontal thing you see is the membrane, and you apply a potential like a voltage across the membrane, and therefore the current flows through the pore, and the DNA goes through the pore, and as the DNA goes through the pore, you keep on measuring the amount of current that is going through the pore. And since the DNA is like DNA has some polarity, charge, so on, like when the DNA goes through the pore, it influences the current that goes through the pore. So we can see that on the right pane. So you see that, okay, like whenever a particular sequence of DNA goes through the pore, you see this raw signal. This is the raw current that is going through the pore, which is measured using basically electronics. And you see that like the current stays flat, then it changes, and then it stays flat, then it changes. So that is because that as the DNA goes through the pore, the amount of current that can go keeps on changing. And it's a quite a complicated process. People have tried to model it, but we don't need to understand like the full details. The only thing to understand is that even for a single base of DNA, like just A, C, G, or T, like one of them, you have like multiple current samples. Like you have many current samples per base of DNA, and that's where like the compression aspect comes in, right? So we can look at it in slightly more detail here. So you see like a typical current sample here. So we have time and we have the raw current value quantized in some way. This is stored in something called a HDF5 file, .fast5 format. And it takes around 18 bytes per base when you store it uncompressed. So this is really big. If you recall, when we were talking about the FASTQ file, it was taking 1 byte per base uncompressed. So this is 18 times bigger than that. And people have worked on compressors. In fact, Oxford Nanopore, the company, they have their own compressor called VBZ, which uses some standard compression techniques, and it can get as much as 60% size reduction over this 18 bytes per base, and it's 30% better than Gzip. But it's really not enough. Like even for like a typical human data, it can take around 1 terabyte of storage for storing just like for one experiment. So this is really big. And again, you might say, okay, like, do we need to store the raw data? So the answer today is yes. In most cases, you need to store the raw data because a lot of the downstream pipeline actually requires you to have the raw data. And also like the processing pipeline is because this is an emerging technology, every few years you get better and better software. So you want to reanalyze your data. And therefore you want to keep the raw data. So actually, when we started looking at this problem, like efficient storage of this raw current data, we worked for I think 2 or 3 months on improving upon VBZ, like getting a better lossless compressor. And we tried really hard. We tried all sorts of modeling, neural networks, and so on. And ultimately, like we were not able to make much headway. And then we started some theoretical analysis. We tried to understand why are we not improving. And we found that the data is really noisy. And actually, most of the compressed size is being used for storing the noise. Like the actual information is relatively small. It's mostly just the noise. So then that like naturally led us to the thought of lossy compression. So lossy compression is just a way of storing data where you are allowed to lose some of the fine details to save space, and hopefully like just losing those fine details don't affect the quality of your data, right? So let's look at some like just a general picture of lossy compression. So we will focus on lossy time series compression because this is just a time series, and we will look at two. Like we use two works. So one called Gzip, which is something we did in collaboration with Siemens, and SZ, which is from another group. Right. So this is the general like framework. So we start with a time series x1 to xn. We compress it, we get some compressed bit stream, which is hopefully very small. And then we decompress it. And we don't get back this x1, x2, xn. We get like some approximate values, some quantized value, x1 hat, x2 hat, so on. And our like the two compressors we use, Gzip and SZ, satisfy something called the maximum absolute error constraint, which means that at every I from 1 to n, you are only allowed to go away from the true value up to some epsilon, where epsilon is like a user-defined parameter. And the higher the epsilon is, the more you are allowed to sort of, you have more flexibility and you can compress it even better because you're allowed to lose more and more of the information. But how do we choose epsilon, right? In our application, what does epsilon like physically mean? So in fact, in our case, epsilon is not the most important metric of accuracy. In our case, what we care about is, let's say I do this, do I lose the performance of everything that I use the raw signal for? Like I use the raw signal for some processing. I use it to get some results. So I don't want to affect the quality of my results just because I do some lossy compression. So for that, we need to understand in a bit more detail what the downstream pipeline looks like, or what do we do with this raw signal, right? So the two like main things we look at is base calling and consensus, and they're like relatively easy things which we already saw earlier. So let's go through it. So we start with the DNA sample, we put it in the nanopore sequencer, we get this raw signal. But ultimately, what you want really is like the ACGT sequence, right? And like we know that the raw signal is determined by the ACGT sequence. So it's just a matter of decoding it back. So that process is called base calling, right? You are calling the bases in some sense. And today it's mostly done using very advanced neural network based techniques. So base calling gives you this ACGT sequences essentially. Right? Like these are your reads. So for each of these raw signals, for each read, you get this sequence. And unfortunately, it's not at all perfect. So here in green, you see the ground truth sequence. And in black, you see the base call. And you see, okay, in many positions, it matches the ground truth. That's good. But in some positions, for example, in the first position, you see you had a T, it got substituted to a A. Here you see you had a G earlier and it got deleted. So in the base call, you don't have anything there. Sometimes you have nothing in the ground truth, but you insert some random GC here, right? So there are a lot of errors that happen in the base calling process, and it has been improving quite a bit over the past few years, but it's still like 5% error or something. So therefore, we use the fact that we are not just reading one part of the DNA once, right? We have a lot of coverage, we have a lot of depth, we are reading the same thing again and again because of the amplification and so on. So what we can do is we can take all the reads that are from the same region of the genome, and we can do some sort of majority vote or consensus. So here we see that, okay, at this position, 2 of the reads had a G, third one had a C. So you could say, okay, like most likely the true sequence had a G here, right? So basically you just take the majority at every position. And here we see consensus errors. So you do this, and so we see again the ground truth in green, the consensus in purple, and we see 3 reads which are used to generate the consensus. So at the first position, 2 of the reads have a T, the third one has an A. So in the consensus, you are able to correct it because you took the majority vote. You saw, okay, 2 were T, so you got a T. So consensus is able to correct a lot of the errors. Whenever like you have a random error in 1 or 2 of the reads, you can correct it on the basis of the other correct reads. But sometimes you have something called systematic errors where all the reads have an error, maybe because the base caller had some issue or something. So if all the reads have an error at a position, then the consensus will also have that error, right? So in all, consensus is much more accurate than the individual reads, but it need not be perfect. So therefore, whenever we do lossy compression, whenever we change the raw signal, we want to make sure that we don't affect the consensus quality and we don't affect the base call quality, because both of these errors are quite important metrics. Right. So with that, let's look at the evaluation pipeline, right? So first stage is compression. We want to generate all these lossy compressed signals so that we can do the analysis later. And so here we start with the raw signal, we do lossless compression. We also do lossy compression. We try several different values of epsilon, right? And we will see how we will use that in the next slide. So you get the original raw signal. That's the signal you got from the machine. And you get all these like quantized or lossy raw signals. Once you have the raw signal, you first do base calling and you compute the accuracy of the base call, right? And we use 3 different base callers, and we will see why we used multiple different base callers here. And then you get the base call reads, this ACGT sequence. And then we can use it to do the consensus and take the majority vote, right? Another thing we did was like, okay, maybe the original data had 100x coverage. You had every position in the genome, you had 100 reads. But what if we reduce the number of reads? Does that affect the quality, right? Like if I use the lossy compression and I use a very low coverage, how do the results look? And so we do that as well. We do subsampling. We basically take only part of the data and then use it for consensus. So the idea behind like using all these different tools, all these experiments is that we want to make sure that lossy compression is not leading to any loss of information that might be useful, right? Like we don't want it to be like, we do lossy compression with one tool, it works well, and then everybody adopts it. And then next year, some new tool comes in, and for that lossy compression doesn't work anymore. So we want to test it as widely as possible to make sure that we are not losing any useful information. We are just throwing away the noise. So in that sense, we also do multiple data sets. We had 1 human data set, 3 bacterial data sets. We used good benchmark data sets where we knew the ground truth. And like the bacterial data sets covered a wide range of like different properties, like different flow cells and so on. For consensus accuracy, we focused on bacterial data sets because it's computationally easier, and also for some other accuracy related reasons. We tested them at multiple subsampling levels. And like this was a really comprehensive evaluation. We tested all combinations of data sets, like different compressor modes, like the different values of how much do you do lossy compression and the downstream tool. And and finally, we also did like some additional analysis, methylation accuracy and homopolymer accuracy, which I will not talk in this today because of lack of time. Right. So we can actually get started with the results. So here we will just focus on one data set. Okay, so this is the base calling accuracy. So there are a lot of axes here. So let me go slowly. So the x-axis is the relative compressed size. So 1 stands for lossless. So 1 is lossless, and you go to the left, you are getting lower and lower compressed size. So the leftmost point is 0.3, which is like 70% lower size than the lossless compression. Y-axis is the base calling accuracy or base call identity. And you see it ranges from around 85 to 92. Now we have 3 colors. So the 3 colors denote the 3 different base callers. So we will just focus on the blue one for now. And then you have like these different shapes. So the square shape represents the lossless compression. So you see it's at the one line. And then you have like the two LF-zip and SZ. And you see like there are a lot of these because we did lossy compression at various different levels of loss. So we kept on increasing the epsilon parameter for the lossy compression. And the more we do lossy compression, the more we go to the left, but the more we also start to lose on the accuracy, which is expected, right? Because after a point, if you keep on like throwing away data, you are going to get bad results. Okay, so the main point here is that even at 35% lower size as compared to the lossless, we are still roughly at the same base call accuracy. So this means that like you can save 35% space without really affecting your analysis, your base call accuracy. And this happens not only for the blue one, it also happens for the green one, the red one. So for all different base callers, the same thing happens, and even for the other data sets. Similarly, we can look at the consensus accuracy, same plot, everything is the same. The only difference is the y-axis is the consensus accuracy now. And you see it's much higher. It's not like 90%, it's 99.9%, because after you take a lot of reads and you do the averaging, your results get much, much better. And here we see that even up to 50% reduction in size as compared to lossless compression, you are still in a very good shape. Like it's still basically the same accuracy. And that's very encouraging, because it means that when you do the consensus, all the errors that are caused due to the lossy compression sort of get averaged out, and you still get very good results. And again, like the same thing holds for like all the different combinations of tools and base callers and so on. And finally, here are the subsampling experiments where like we do no subsampling. So the original data, we subsample it by 2 times, 4 times, 8 times. And you see like as we keep on subsampling more and more, the consensus accuracy decreases, which is expected because you don't have as much data now. So your results are likely to be worse. But again, like if you go from right to left, you again see that up to a lot of lossy compression for every, like if you look at just one color, you see you can still reduce the size quite a bit without losing any useful information. You still get the same consensus accuracy even after, like even at all levels of subsampling essentially. Right, so let's summarize. So we show that we can achieve 35 to 50% reduction over the best lossless compression. And if you recall, lossless compression was already 60% better than uncompressed. So this is really huge reduction with negligible loss in accuracy. And we get consistent observations across data sets, coverage, downstream tools, and so on. This is highly practical. It's not like some theoretical idea. LF-zip in particular simply reduces the data resolution. Like instead of 1.111, you are just doing 1.11. So it's like a simple uniform quantizer. And one can potentially just adopt it at the nanopore sequencer device, because the device has some level of resolution. You can just reduce the resolution at the device. This is the first work on the topic. Much remains to be explored. In particular, we just use the models which are designed for the original data. If we retrain the models for this lossy data, maybe we can get even closer to the original results, maybe even better than the original results. We will just use general purpose compressors for this data, but maybe we can develop specialized lossy compressors specific to nanopore, and maybe they can give even better results. Our evaluation on human data was somewhat limited, partly because we didn't have very good benchmark data sets, but we hope there has been a lot of progress, and we hope in a few years we might be able to do a much deeper study even on human data sets. And all the evaluation scripts, data, plots, everything is available on GitHub, which we hope can allow others to do similar analysis for their own favorite lossy compressor. So I will briefly mention some publications. So the works I talked about today are published in bioinformatics. So these are on the genomic data compression. So we had the work on the lossy compression that I mentioned second, and we had 2 papers on the spring work. One topic, one main focus of my research, which I didn't talk about today, is the storage in DNA. So this is sort of complementary. Here we are using DNA as a storage medium for storing all the data the world is amassing. And so we are currently working on a journal paper, and we had 2 conference publications on this. So this is mostly about error correction. And finally, there have been a few works on time series compression and a few really fun projects on multimedia compression. So the time series compression is in collaboration with Siemens, and it's the LF-zip work, which I briefly mentioned earlier. And the multimedia compression works are with high school summer interns, and they have been really fun projects to work on. So all these were published in DCC conference. Right, so I will quickly wrap up with thanking everyone who has made this possible. So first, I would like to thank the exam committee, Professor James Zhao, who is the chair of the committee. Thank you. I want to thank Hanley and Mary, with whom I've had the pleasure of working on in the DNA storage in DNA project, and I've learned a lot from both of them, and I've learned a lot about their respective fields of expertise. So thank you, Hanley, Mary. And thank you, IFER. So this is the first quarter I'm really working with IFER in the context of the class that Tsachy mentioned, and it has been a really fun experience. I want to thank Tsachy, my PhD advisor, for his constant support in research and in life. And yeah, I think most of all the opportunities I got, I think almost all of them were due to Tsachy. In terms of exciting new open problems, research areas, even a lot of mentorship opportunities. Working with him has really broadened my perspective through mentorship, TA-ship, and exciting research. So thank you, Tsachy. Next, I would like to thank Kedar, who has been really a great friend and a collaborator for almost all of my works, which I did during the PhD. I also want to thank Pulkit and Jai, also great friends and collaborators for some of the projects. So with Kedar, Pulkit and Jai, all of whom are from IIT Bombay, where I did my undergrad, I never felt that I was not at home. Yeah, it felt like home with all three. I want to thank the entire Weissman group, who are many of whom I've had the pleasure of working with, and yeah, it has been really fun. There are some of you I haven't met in person yet. I hope to do that very soon. And of course, all of my research has been really great collaborators who have introduced me to new research areas, exciting open problems. They have mentored me. I have had the opportunity of mentoring some of them. So yeah, I want to thank all the collaborators. It has been a real pleasure to work with all of you. I want to thank the EE department at Stanford, who have provided great support throughout and have really made this process so smooth. And finally, I want to thank my parents and my brother Siddharth for all their love and support. Thank you. And so I will end with thanking all the audience for listening patiently, and I will take any questions.
So I think I'm still in charge of this part before I hand it over to James, the closed session. So maybe I'll start with the question we had on the chat. How is the accuracy measured? Just by repeating the process?
Oh yeah, so I think this was in the earlier part, I think.
Yes, I believe that was in the first half.
Yes, yeah. So I think accuracy is just yeah. So for measuring the accuracy, you need to know the ground truth sequence, like what is the true sequence, right? So after you do the sequencing, you get these reads, and we have a pretty good idea because all the human genomes are really similar to each other, and there are some human genome sequences which are known to, like there was some particular person called NA12878. So that person's genome we really know very well, and we have the sample from that person. So you could just sequence that person's genome using whatever sequencing technology, and then you can see, because you know the ground truth sequence, you can just compute the accuracy by comparing your read to the true sequence. Okay, so there is.
How do you obtain, oh, sorry. So there was another question here in the chat. How do you obtain the consensus between the nanopore reads? Thank you and congratulations.
Thanks, Guillermo. Okay, so I think we had a slide. Let's. Yeah. So in terms of consensus, yeah, good question. Yeah. So so the first step is usually alignment. So you have a reference genome. You align the reads to the reference genome so that you know roughly which reads are in the same part of the genome. That is often followed by a local assembly process where you take all the reads that are in the same region of the genome and you do an assembly process. You don't do like a comprehensive assembly for the whole genome. You just focus on this region of the genome. And like today, like as with everything in life, people have developed like the final stage is done using neural networks. So they have trained a lot of trained using a lot of data. So you just put in like your reads into as the input of the neural network, and the output is the consensus sequence. Yeah. So yeah, so that's like the standard.
Bernd?
Yeah, I was about to type a question, but I thought it's maybe easier to just speak up here. And so first, congratulations to getting to this point and to presenting this nice and interesting work. You know, in the genome, there are always triplets of base pairs, right? Each triplet encodes an amino acid. There are only 20 amino acids, so there's some natural redundancy in that right there. And I just wonder how that manifests itself in the algorithms that you use. I mean, it seems like everything you do is basically oblivious to this, you know, triple base pair structure, and of course, you know, in principle, you could even do some denoising, knowing that certain triplets are not allowed, right? It's
yeah. So that's a good question. So I think one
in fact, I believe even even in the cell, that type of repair is done, right? If you do have some, you know, if you have some I mean, you have enough redundancy in the genome that the genome will repair itself if there's some damage.
Yeah. So I think that's a good point. Okay, so I think in terms of the genome repair, I think it's not purely based on the genome, like because when you duplicate like when you make a copy of a genome, you have the original as well. So the repair mechanisms often work by like comparing the original with the new copy.
Yep.
And in terms of like the codon thing, like the three bases give one amino acid. So I think so interestingly, like since I'm from an electrical engineering background, I think for the first 3 years of my PhD, I didn't even know that. So a lot of this work was like not done, not knowing that. So I guess like the real answer there is that most of this work is about whole genome sequencing, where you're sequencing the whole genome. But the coding part of the genome is just 1% of the entire genome. And that's where all these like 3 base per amino acid and all that comes in. For most of the genome, that doesn't happen. So yeah, but it's an interesting question. I can look it up.
I mean, the question is, does your lossless compression algorithm pick up this redundancy? There are just certain sequences that cannot occur, and it's all on a very short range basis, right? I mean, there are no long range things. I mean, it's kind of on the most elemental level. So I mean, it could be that your algorithms pick it up. I just don't know, right? So.
Yeah, that's a good point. So actually, like that's why we used like the BSC compressor I mentioned, like the final stage of our pipeline. So it is like a really good compressor. It works in large blocks. It uses BWT based techniques to do the compression, and we found that it actually does quite a bit better than Gzip. And I think that's because like it can capture all these like effects, like all these redundancies, all these statistics of the genome much better than Gzip.
Thank you.
So I think we're going to end with CZ's question.
Sure. My question is a non-technical question about Spring. So I appreciate that you were very eager and enthusiastic about having it adopted as a practical tool and making sure that it was usable. I'm curious, what did you find? What did you feel was the biggest challenge in getting it adopted, or in seeing it adopted by the sequencing community?
Yeah. So I think okay. So I wouldn't say like I think still people use Gzip because ultimately I think like unless it's standardized by somebody, it's really hard to get people to really adopt it. But we did see like a lot of people contact us, and some of them actually told us that they are using it on their data. So I think one big thing was that it should be very easy to install. Like it should not require like a lot of programming expertise to get it running. And that is why we put it on Conda, where you just do like Conda install Spring and it installs Spring on your machine. So that was one big part. Another big part I think was that we made the lossless mode, even though Spring has so many lossy modes, we made the lossless mode the default mode, because for a lot of practitioners, they want to retain their data. Like even if the data might not mean anything for the analysis, like people still want their data in case it becomes important in the future for some reason. So they want to store the data as it is. So that was the second thing, like making lossless compression, lossless compression the default. And finally, it was, I think, about computational requirements. Like Spring does take up a bit of RAM and time, but we did try to optimize it as much as possible, because like some of the competitors were really too slow. Like even we didn't want to run it when we were doing our experiments. So yeah, so those were some of the factors that came in for making it like more accessible.
Thank you. I think we're going to wrap this up by thanking Shubham again. And I'm going to hand it over to James for the closed session.
Yes, we'll go to a separate Zoom for the closed session. Thank you, Shubham.
Thank you.